Therefore, some countries have been interested in settling organic agriculture in many parts of the country. This is done by converting organic waste and agricultural products to organic fertilizers, especially in countries characterized by drought due to lack of rainfall, scarcity of vegetation, and high temperatures. In desert countries, lack of intensive cultivation methods resulted in a decrease in biofertilizers and low organic matter, resulting in reduced soil fertility. http://www.fao.org/organicog/oafac/oafac1/r. From this point of view, one of the main objectives of this study is the use of olive press cake, OPC, from many olive mills spread in Juf region in the northern part of Saudi Arabia, as biofertilizers in organic agriculture. The number of fruitful olive trees in such area was estimated to be more than 15 million trees, produced more than 12 million liters of olive oil. The agricultural land of the city of Sakaka and its suburbs, belonging to the Juf region, of the northern part of Saudi Arabia is characterized by the lack of suitable physical, chemical, and biological properties. Therefore, we have considered using enormous amount of residual OPC in raising efficiency of agricultural soil through a number of successive researches in this field. As a result of the huge quantities of the remnants of the process of refining olives, large quantities of waste are formed with other pollutants from the wastewater of these processes. These pollutants are of big environmental problem because of their high organic load. The addition of OPC to agricultural soil increases organic matter and inorganic elements essential for plant growth. By contrast, the application of OPC to the soil causes phytotoxic properties due to the high content of phenolic compounds. Generally, the mushroom fungus, Pleurotus, can grow well on organic residues containing lignin and lignocellulose, since these fungi are able to analyze these substances and produce simpler, more nutritious residues, and more benefits to plants. Previous studies indicate that the first stage of mycelial growth of the mushroom in some terrestrial fungi is to be biomass, followed by a decrease in the concentration of harmful phenolics, which turns waste into organic residues enriched and useful for agricultural soil. Analysis of components of OPC is found to contain ash, lipids, minerals, polyphenols, polysaccharides, proteins, sugars, and tannins. The concentration of phenolic compounds reaches up to 10 grams/L, which causes high plant toxicity and antibacterial properties. We have benefited from these data that we designed researches based on the use of certain fungi in the withdrawal of high phenols of OPC and then converted it into organic fertilizers added to agricultural soil. Useful fungus of Gliocladium roseum, Pythium oligodendrum and Trichoderma hartzianum, and the mushroom of Pleurotus ostrichusweri used in this respect. It is well known and noted through many previous studies that G. roseum, P. oligodendrum, and T. harzianum have a long history of biological control of many fungal plant diseases. The mushroom of P. ostrichus mushroom is also known for its high nutritional value and a good source of protein for many people. Therefore, the use of G. roseum, P. oligodendrum, and T. harzianum has more than one benefit. The first is the withdrawal of the high concentration of phenolic materials from OPC to be suitable for agriculture. The second, that these fungi are important in the biological control, constitutes a wonderful medium to exist within these organic fertilizers. It is worth mentioning that G. roseum, P. oligodendrum, and T. harzianum have the ability to secrete substances similar to plant hormones, auxins, that cause increased vegetative growth and productivity of plant crops. The overall aim of this study was to use P. ostrichus mushrooms as well as G. roseum, P. oligodendrum, and T. harzianum to grow on OPC to benefit from the productivity of mushrooms and make it suitable as a biofertilizer. G. roseum Ju 121, Juf University, Saudi Arabia, P. oligodendrum Ju 221, Juf University, Saudi Arabia, and T. hartzianum Ju 321, Juf University, Saudi Arabia, were isolated from 25 agriculture fields distributed in Hoa village, Sakaka, 29 degrees 48.6 in, 40 degrees 26.27 inches, Juf Governorate, located in the northern part of Saudi Arabia. P. ostrichus muigri 1102, Egypt, fungus was kindly obtained from the Ministry of Agriculture, Egypt, as a ready spawn grown on grains of sorghum. Afterward the spawn prepared by subculturing the fungus on the medium of malt extract agar, was used, figure 4. Spawn of mushroom off. Ostritas grown on grains of sorghum. OPC was obtained from an olive mill located in Sakaka City, Juf, Saudi Arabia, and used spontaneously after sterilization by autoclaving. Experiments were performed in a glass house, and 2 treatments were used, control plus 5 replicates. Subsequently, results were statistically arranged and all treatments were compared using Duncan multiple range test. 95% vermiculite and 5% gypsum were the only components of the control. Treatments were prepared as 95% olive press cake and 5% gypsum, dry weight. Gypsum was added to each treatment and mixed thoroughly and then placed in a cloth bag. Autoclaving was done for 2 successive days at 121 degrees Celsius for 1 hour and left 3 days before use. The glasshouse was disinfected using sodium hypochlorite. The medium was replaced in big plastic bags in order to allow the manipulation of mixing the spawn with the substrate by thoroughly shaking. Subsequently, medium was inoculated with 5%, dry weight, spawn of P. ostritus. The bags were sealed tightly with a strong thread and punctured with a sterile metal screwdriver. Substrates were incubated at 20 to 25 degrees Celsius, under 80 to 95% RH, humidity in the dark during starting days until the emergence of white mycelial growth. The colonized substrates were subjected to a cold shock at 5 degrees Celsius for 48 hours to stimulate the emergence of first flush. It is worth mentioning that ventilation was very important during the fruiting period, therefore, the upper side of the bags was opened. Precautions must be taken for the temperature to be around 25 degrees Celsius and the relative humidity was between 80 and 90% by watering the bags twice daily and placing vast water containers on the floor. Basidiocarps, fruiting bodies, of P. ostritus had been collected when pili were matured and just before started to curl up. Residues attached on stipes of mushrooms were gently disposed of by wiping them with a tissue paper before weighing. After harvesting mushroom, the average weight of singular basidiomata calculated as the quotient of the total weight of fresh bodies collected by their total number, the average production for each parameter and diameter of the pili, and the average diameter were measured. Olive press cake, OPC, was collected from an olive mill, Aluba, Sakaka City, Juf, Saudi Arabia. Fungi were developed and preserved in potato dextrose agar, PDA. Part of this work has been reported elsewhere. Potato dextrose agar discs containing fungal growth were used for OPC inoculation and subculturing, as well. Incubation procedure was performed in 1-liter Erlenmeyer flasks, each containing 200 grams of OPC and 150 milliliters distilled water at 28 degrees Celsius for a time course of 1 to 4 weeks, figure 5. A. OPC and water in flasks after sterilization in the autoclave. B. Flasks during inoculation by fungi. C. Flasks after inoculation. D. Fungal growth after incubation for 2 weeks at 28 degrees Celsius in the dark. The total phenolic contents of OPC were estimated according to the method of via tannic acid as a standard, and expressed as grams per kilogram of OPC. Analyses were done for each treatment before and after growth of tested 4 fungi within 1 to 4 weeks. The OPC before and after culturing with each of the tested 4 fungi was analyzed for their appropriateness for growing seeds of E. sativa. Quantities of every 100 grams of OPC were added to plastic pots. 50 E. sativa seeds were distributed on the surface of each pot containing tested OPC. Pots were incubated in an illuminated growth cabinet at 25 degrees Celsius with 12 hours photoperiod, 91 mol m-2s-1. Emergent seedlings were counted in the course of 5 to 20 days. Data were analyzed using one-way analysis of variance, ANOVA, through Minitab statistical software, version 12, unless elsewhere mentioned.